Hey there, everyone. We'll go ahead and let people start filtering in. As you're getting situated, I'm going to go ahead and pull up a poll, fill that out at your leisure. It'll just go ahead and let our presenter, John, know what to kind of speak to today. So I'll get that started. And for those of you who are coming in now, it is great to see you all. We're getting excited for today's webinar. You may see a poll open. That just gives us more information about what to speak to for this webinar. So please let us know, what you're looking for, and we'll go ahead and kind of work that in. Well, before we dive in, I'll go ahead and do just a little bit of housekeeping right off the bat. First of all, thank you all so much for joining us for two simple ways of breaking down cellular respiration. I'll turn the webinar over to John here in just a moment, but first and foremost, the recording of this webinar will be available on our website. We will also be sending out a follow-up email with a link to the recording, so you'll be able to watch or share with anybody in your circle. Make sure that you can get the word out. And we would love to answer all of your questions during today's session. So please feel free to use the chat section on Zoom to ask your questions. I'll keep an eye on this. John will as well. If we happen to not get to your question today, we'll make sure we follow up with an answer. So with that being said, I'm going to go ahead and end the poll and we will dive in. Thank you all.
Great. Terrific. So that was kind of, I just looked at the poll numbers and that was kind of the assumption that I had, is that most of you, I mean, we do have some college folks here. That's great. These sensors are great for college. We have a bunch of high school folks here. That's great. These sensors are perfect for college, for high school as well. And it looks like most everybody's interested in the sensors themselves, not so much the software, special features for the software. We are going to cover software because that's how the sensors are actually used. But that was the focus that I was going to focus on here, is focusing on actually three really great sensors for studying cellular respiration. It's really two experiments, but you can do one experiment in two different ways. One experiment, you can look at CO2 gas, and then you can look at CO2 gas with O2 gas, and then you can look at dissolved oxygen. So you're going to meet me. My name's John Melville. You'll meet me here in a minute. I'm going to go through all three of these sensors and the experiments related to them. It's going to be a hands-on demonstration, so I'm going to actually show you how to do the first part of each experiment. Please feel free to ask any questions in chat. Like, just throw questions in chat. You can throw questions in chat right now. Yeah, great. So someone asked it, that's great. Someone just asked a question like, hey, I've got issues with the O2 gas sensor. I'm gonna cover that later. The O2 gas sensor is one of those things that you probably want to calibrate it when you use it. And don't worry, we'll also cover like how do you view the webinar today as well. But that's perfect. It could be, the other thing about the oxygen gas sensor we'll cover later is you may, if it's old, you may need to get it serviced, and we can talk about that. The other thing I want to talk about, let me introduce myself and then we'll talk about some stuff. So my name is John Melville. I'm the director of the biology department. I'm also the biology staff scientist at Vernier. I have a BA in biology and a BA in psychology from Sonoma State University. I got my PhD at Oregon State University in zoology. I like to say that I got my PhD at what we call the AME-BME boundary, which means it was like right at the cusp when we went to the molecular era. So I'm a broadly trained general zoologist. I've taught lots of courses from marine biology, general biology, introductory neuroscience, general biology, human physiology, etc. I did my postdoctoral fellowship at the University of Oregon, also in neuroscience, but most important for this group, I'm a former educator. We like to say that Vernier is for teachers by teachers. And I used a lot of Vernier equipment in my classroom for both majors and non-majors courses in biology, including neuroscience, but definitely general biology. And my focus was really on active learning. I wanted all of my students to collect data and analyze data. And I also focused a lot on inquiry. I tried to make every experiment that my students would do inquiry-based, or find a way for them to even do what we would now call guided inquiry. But I wanted them to do experiments and I wanted them to be excited about science. So feel free to also ask me any questions you have about teaching the classroom, things like that. It's been a long time, but I know a lot about how these sensors can be used in the classroom, what they're good for, and then also like what I wouldn't use them for. And we can talk about that. All right, so just to go over some just basic features before we jump into how everything works. There's a chat feature. Like I said, ask all your questions in chat. Someone will be answering your questions there as we go. It'll probably be me if I see them there, or if I just continue talking, Dylan will probably, jump in and say, "Hey, John, here's a question." I mean, don't worry, there should be time at the end of this for you all to ask questions, and we can have a bigger discussion at the end. I am going to try to do data sharing later. So if you want to find graphical analysis on our website, you can download it if you do not already have it. Just make sure it's the latest version if you already have it, and update it if necessary. Don't worry, I can share data with you. This GA Pro key will give you access to GA Pro. It's ZXKSFKFLE. I can put that in the chat later as well if you want. But all that you need to do to go there is, if you go to our website, just tap on downloads. You will see Vernier graphical analysis. You can just go to download. And it will tell you what operating system you have, and you can click there to download graphical analysis. If you're on a Windows computer, you can download the native app for Windows. There's one for Mac. The Chrome OS, if you're on Chrome, that'll take you to the Chrome store. If you're on Android or iOS, you need to go to those stores, but this should take you to where they're located in the store to get them as well. So graphical analysis can work on all of those. There's a question. Oh, thanks, Dylan. Dylan just put in the GA Pro key. I can show you how to put in the GA Pro key later too, if you want. It should be down at the bottom, but we could talk about that. So let's go back to the slides. So Dylan put in the GA Pro key. All that you need to do is when you launch graphical analysis, the first time you launch it, you should see this down here, which says activate additional features with your license key. Just put in that GA Pro code, and then you have GA Pro. So that's the GA Pro key that you want to put down there. Now, just to let you know, everything that I'm going to do, all of these experiments, do not require GA Pro. GA Pro is really nice. I'm going to show you some GA Pro features because you can data share. You can, there's lots of more advanced features in GA Pro. There's also videos and experiments that are in GA Pro that I'm going to be using in this webinar to show you how the experiments are done after I show you some of the basics. But you don't need GA Pro to use our sensors in the very basic way, collect data, and do most of these experiments. It's just a really, really nice feature. GA Pro is a lot more like Logger Pro. GA without the Pro is a lot more like Logger Lite, is the way we like to think of it. And there are 1-year site licenses, 2, 3. You can even get a 5-year site license for GA Pro if you want it. All right, so do we have any questions? What I'm going to focus on next is mostly going to be on sensors, 3 sensors that can be used for looking at cellular respiration, and then different ways to look at cellular respiration. And I'm going to focus on 3 sensors: the Go Direct CO2 gas sensor, the Go Direct optical dissolved oxygen sensor or probe, and the Go Direct oxygen gas sensor. Now, the CO2 sensor and the O2 gas sensor are both gas sensors. They're used in air. You can't put them in water. The dissolved oxygen probe you can put in water. And I'm just going to show you a different lab associated with it. All right. So let's just talk a little bit about Go Direct sensors. Now, Go Direct sensors, you may have the older sensors, and that's fine. Don't be worried that I'm talking about Go Direct sensors. All of the tricks that I'm telling you, everything that we're going to be doing, all of these experiments, there are also experiments written for our older, what we call LabQuest sensors. They work great. And a lot of the tricks that I'm going to be showing you, same tricks that you need to know to get them to work. I just wanted to let you know that the Go Direct sensors can be used with the USB cable. A lot of people call them wireless. They're not just wireless. They can be used with the USB cable. They can be also used wirelessly using BLE. They also, really importantly, have many different sensor channels. And a lot of people don't get that the GoDirect CO2 sensor is not just a CO2 sensor, but you can measure relative humidity and you can measure air temp all at the same time. The GoDirect optical dissolved oxygen probe can measure percent saturation. It can measure dissolved oxygen in milligrams per liter. It can also measure water temp simultaneously, and it has a salinity correction. So the GoDirect sensors are really great because you don't need an interface. The cost to get going is much less. They work on literally any platform. So they can work on iOS, Android, Chrome, anything. But they cannot be used with Logger Pro. They have to be used with graphical analysis. The one caveat is they can be used on our LabQuest 2 or LabQuest 3, either with the USB or with Bluetooth. It's just that you could not use them in Logger Pro. But once again, don't worry, all the tricks that I'm going to be talking about and showing you, etc., all of these tricks are also relevant if you have our older sensors. And all of our older sensors as well can be used in Logger Pro or with graphical analysis if you have the proper interface. All right. So don't forget to just go ahead and ask questions in chat if you want to. All right, so the two big things I'm going to be talking about with you are showing you the tricks on how to look at two simple ways of breaking down cellular respiration. One is going to be looking at peas. So we're going to be looking at seed peas. And I'm just going to be showing you all the tricks to make sure that you can get really good CO2 data for just with the CO2 gas sensor. And then also more importantly, with the CO2 gas sensor and the O2 gas sensor. And then I'm going to show you a really neat experiment that looks at aerobic respiration using yeast and water, which is really, really simple. And I think it's a great lab. Don't forget, you can ask questions in chat at any time. All right, so where are these experiments coming from? The experiments that I'm showing you come from two books, Biology with Vernier, and then Investigating Biology through Inquiry. Biology with Vernier is our go-to book. It has more cookbook labs. It's just an amazing resource for anyone that's teaching biology. It has over 32 labs in it. It covers a very broad collection of experiments. It's a great resource. But they are more cookbook. So it's more, this is what you do, this is how you analyze. This book is perfect for on-level biology and also I would say like non-majors biology at the college level. Perfect. I use it a lot to show people great experiments that exist in it. If you're interested in inquiry, and some of you did ask about inquiry, our inquiry biology book is better. But inquiry is complex. You can do lots of different experiments. Students can do lots of different experiments. And this book has both guided inquiry examples and open inquiry examples. It's an amazing resource if you're going to be teaching, say, like AP inquiry style labs, or you're going to be doing college inquiry. Most importantly, because of the teacher section, which will show you certain things that you can do, could do, and even shouldn't do. All right. Great. Dylan dropped both links to both books in the chat. By the way, I'm one of the quote unquote authors on Investigating Biology through Inquiry. So I also have done all of these labs. How much have these manuals changed since they first came out? They really haven't. Neither of these books have changed that much since they first came out. The one big thing that has changed is they have both been updated for graphical analysis. So there are instructions in them for graphical analysis. But that is a great segue to the following, which is if you already have these books, you can get access to them by just logging in on our website. So if I type in my name here, and then let's see. See if I remembered my web login. It's thinking, so that's probably a good sign. There we go. So if you create a web account or get a web account, it'll show you all of your software installers. No. Subscriptions, but most importantly, your lab book electronic resources, right? So I have access to all of these books. So if you already own one of these books, you don't need to buy a new book to get the updated graphical analysis instructions. They're already there. Just go to the website, create an account, log in, and download the updated instructions. And that's another important thing, which is you don't need to buy one copy for each teacher. The way that this works is it's one book if you buy a book, it's a site license for a college department or for an entire like high school. So if you buy one book, maybe you bought the book and you want the paper copy. Well, once you log in and get it verified, that it's a purchase and you get it in your account, anybody at the high school that wants it, that teaches in your subject area, well, actually anybody can get access to it because we just look at your email. So any questions on that? Like, they're really affordable too. They're only like around 48, or they're less than $50 each lab manual. All right, so let's go to a live demonstration. Let's talk about the basic experiment, which is cellular respiration. Let's just talk about the GoDirect CO2 gas sensor. And really what we want to do is we just want to show that CO2 goes up, right? So let me go over here. I'm using an IP EVO camera, which I really like. These are all my toys that we're going to be using today. Let's just move these out of the way a little bit. There we go. So you should see right here, we have an Nalgene bottle that comes with the CO2 gas sensor. And if you look at the bottom of the CO2 gas sensor, you can see right here, there's a USB port. So you can plug it in over USB, or you can connect wirelessly to graphical analysis just by tapping right here. It'll start flashing red. That means it's broadcasting. And then while we're here, what I'm going to do is I'm just going to take about 25 or so peas. Doesn't really matter. I'll just take a little handful here. I'm going to blot them. Then I'm going to put them in the Nalgene bottle. And these peas have just been sitting in water for 24 hours. There we go. So I got seed peas. I put them in water last night. They've been sitting for 24 hours. So the sensor's getting ready to go here. Let's go back to graphical analysis. I'm just going to say new experiment. I'm going to choose sensor data collection. And I'm going to connect wirelessly. And I see this number GDX-CO2 4Z8. Let me just make sure that's the right one. Nope, it should be GDX-CO2 389. So let's just search for CO2. Ah, there we go. GDX-CO2 1389. I'm going to connect. There we go. And you can see right here that I can open up different sensor channels. So if I wanted to look at temperature and relative humidity, I could. All right, so to do this experiment, let me just make this a little bigger for you. I'm going to go to app preferences. I know that's wide enough, 1.5. I'm just going to collect for 300 seconds here. Then I'm going to hit collect. Now, one of the big tricks that I use whenever I'm doing this experiment with students or showing teachers is, in a lot of cases, right, if we're doing a biology experiment, it's slow and it takes time to see a response. You can see that the CO2 level is going up here, but for most students, that seems kind of boring, right? They're going to be waiting 5 minutes. Down here in the bottom left-hand corner is the zoom to all data, and that's your friend. Because as soon as you see CO2 going up, you can tell your students, hey, tap on the zoom to all data button. And then they'll be able to see that it's already going up. And now another trick that I want to show you here, if we go back, you'll see that I put this CO2 sensor on its side. The instructions tell you to stand it upright. I put it on its side. There's a reason for that. And it's because CO2 is heavier than air. And if I lay it down flat, it's going to move faster. So you see a faster response. It'll still work great if you stand it upright, but you're going to get a better response if you lay it flat. All right, questions while we're waiting for this? Oh, big thing. Well, let's go back and let's talk about that experiment. Why do I like this experiment so much? We'll come back to this in a minute. I mean, all that I'm doing here is I'm using this to measure the rate of respiration in peas, which could be any seeds, could be beans, could be anything. There's other experiments that you can do with it. You can look at respiration and photosynthesis from leaves. Spinach leaves work great. The CO2 sensor is great for that. You can measure rate of respiration from yeast when they're consuming different sugars, works great for that. You can also use it for soil respiration. And don't forget that it has separate channels that can be used to measure air temperature and relative humidity. Now, the reason why I love this experiment is because what is the number one misconception students have about plants? When I was teaching, the number one misconception is they think they only have chloroplasts, right? They think, oh, plants have chloroplasts. They don't have mitochondria. And so they think cellular respiration doesn't happen in plants. They think that it's just photosynthesis. And of course, that's wrong. You know, plants have both mitochondria and they have chloroplasts. So they have to do both. And so peas, the reason why I love the peas is they're green. And you'd be surprised how many students are going to sit there and say, oh, wait, these are peas, they're green, they should be photosynthesizing, what's going on? And that's a great time to talk about cellular respiration plants, and that, you know, plants do both. Animals just do one thing. They just do cellular respiration. Well, they also do aerobic respiration, but we just do respiration, right? They can use other compounds, etc. But plants, the big thing is they do both. They do photosynthesis and respiration. All right, so let's go back to the data here. You can see it's been about 200 seconds. And the other thing I love about this lab is I get this great response, right? It's just works almost every single time. And all that you have to do is have about, 20, 30 peas. I can get this experiment to work if I've got the peas in water for just like an hour. But if you just let them sit overnight, you're going to be fine. If you're going to let the peas sit for several days, like you want to reuse them, always wash the water out every day, put new water in the day before, the night before, and then let them sit and you can use them. That's actually a great experiment. We've had people that have examined as an inquiry experiment how the respiration rate changes, over days. You can use dried peas, right? The classic experiment, I'll show you that next. Let me hit stop here. All right, so how do you look at the rate of respiration? Well, you just slide across the range. You go down to the bottom left corner where it says graph options. And you apply a curve fit, and it's going to default to linear. And there's your slope, 3.846, which is your slope or your respiration rate per second. Right. If you want to make this respiration rate per minute, just multiply by 60. All right, great question. Someone led into that. Do you use dried peas, or what we call non-germinating peas? Yes. So let's take a look at a new experiment. Let me disconnect the sensor for this, and I want to show you a really cool feature I'm going to remove the peas, disconnect my CO2 gas sensor. And I'll show you a really cool experiment, this same experiment that exists in GA Pro. So if you go to GA Pro, you can go to See Experiments, and this experiment exists there with video. And so I can go to Cellular Respiration. And what you'll see here is that there's a video. So this is the exact same video that you just saw, right? But I've inserted this video. And then all of the data collection is here, all of the 3 different runs. So there's germinating peas, non-germinating peas, and then cool germinating peas. And you can easily see from this graph, right, that germinating peas, the slope is much bigger than cooled peas. And then if you have non-germinating peas, you barely have any respiration rate at all. So that's how the classic experiment is done, is you look at, non-germinating peas, germinating peas, and then you also do an experiment where you look at cooling the peas down. I always recommend doing germinating peas first so students can see a response. Otherwise, it's really boring if you do non-germinating first, right? And this experiment lives in GA Pro, so if you wanted to show students this experiment, you can. If you have GA Pro, you can enable replay, and I'm going to pick the experiment that I want to see germinating. It's going to play back at 3 times normal speed. And so you could tell students, hey, open this up, look at it. If they, for some reason, weren't able to do the experiment or don't have it, you can basically use this as your backup, and then they can analyze it. So it's just going through the video, showing me putting the peas in there. That's me wearing gloves. That actually is my downstairs bedroom during COVID when we made these videos. Then it says wait 60 seconds. That's the play button. And then you should be able to see down here the data coming through. This is real data. So this is the real data from that experiment linked to that video. So we didn't fake any of this. This is the real data. So questions? Pause that. All right. So that was, let me just go over what I just showed you, or we were talking about. So the first thing that I did there when I showed you the CO2 gas sensor is we talked about in-person learning, right? That's me just showing you how you can collect data. The student can collect data. I was collecting data. There is a way to data share, which we can talk about later, but students can also just collect the data themselves using the probes on their computer. There is also asynchronous learning where they can take a look at the video. So what I was just showing you is the video that exists for that experiment in GA Pro. Oh, I already did the live demo, so I jumped ahead of myself. So that live demo was that. So I was just showing you here the remote lab that it lives inside of it. And students can analyze it, they can take a look at it, you're not going to break anything by looking at that file. They can even save it elsewhere and analyze it. All right, so I jumped ahead of myself. No worries. So there's these experiments that live in video, experiments with videos that live in Graphic Analysis Pro, and I just showed you that one. All right, so next experiment. The next experiment is actually the same experiment, but it's actually looking at oxygen gas at the same time. And this is what some of us call, that the perfect experiment. We want to show CO2 going up and O2 going down, and we want students to see that oxygen goes down while CO2 goes up. And this experiment tends to work really well with peas. There's a couple tricks to it that I want to go over with you. They're in our instructions, so they're all there, but I want to make sure that you hear me say the tricks that we use to get them to work. So this is exactly the same experiment. It's looking at cellular respiration with peas, but we're going to use the CO2 gas sensor and the O2 gas sensor to see CO2 levels go up and O2 levels go down. So we're going to be measuring the rate of CO2 production and O2 consumption in peas, or these seeds. Now, the key thing is to make sure the O2 gas sensor is calibrated And then what I say is make sure you use 60 instead of 20 or 75 instead of 25, or just fill the entire bottom of the chamber. You also need to use a different chamber, and then you also want to make sure both sensors are set to the same scale. There's a reason for that. The reason why you want to set them to the same scale, so make sure they're set at PPT, parts per thousand, and then set up 2 graphs, is really because you want to make sure that you can see a response. Now, what I like to say is there's very little CO2 gas in the environment. It's around 400, 410. I'm speaking right now in a room. It's probably a lot higher than that because my exhaled CO2 is around 40,000 or 50,000 ppm. But the CO2 gas sensor is really good at measuring CO2. So any change that it sees, there's no CO2 around, there's no background. So it's going to be really good at seeing a change. We have the opposite with oxygen. There is around 21% at sea level oxygen gas in air. So if you want to see a change in oxygen gas, it can be really hard because you have this huge background of 21% and you're trying to see little teeny changes with some P's in a chamber. So that's why it's good to change the units and make it not percent, but parts per thousand or parts per million even, just so that you're more likely to see a response. All right, so let me show you that experiment. We'll do a live demo here. So I'll go back to my setup. I will not be using this Nalgene bottle. I'll be using this Nalgene bottle. This is what we call a BC250. It just has a grommet in the top. And it has a, it's just a Nalgene bottle with a grommet. The reason why we have it this way is because the oxygen gas sensor, this is the oxygen gas sensor, you always want it standing straight up and down like that. You wouldn't want to stick it in the side. So the CO2 gas sensor will go on the side, O2 gas sensor goes on top. And I like to say the cap for the bottle is perfect for just holding it still. Like that. And then what I'm going to do here is I'm going to turn both sensors on. And then I'm going to take out some P's here. I'm going to blot them dry. And as you can see, I'm just going to use a lot more P's. The reason why you want to blot them dry is because relative humidity can also interfere with your oxygen gas measurement. So you don't want them super dry, but you just don't want them super wet. All right, so now I'm gonna just move those P's into my hand here. I'll just put a little more in there. There we go. All right. So let's go back to graphical analysis before I set this up, because remember I said something about making sure the O2 gas sensor is calibrated. So let's go back to GA. I'm going to do File, New Experiment. And I'm going to connect my O2 gas sensor. Should be O2 N7. Yep, N7. Oh, got lost there. And connect over USB really quick, just for fun. There we go. So you can see what I did there is, it's almost like I did that on purpose. I've just connected it over USB. And you can see this is also a multi-channel sensor. It's also got O2, this is a rapid temperature compensation. Most people probably won't need to use that. And then it also has a temperature sensor on it. Well, what's great here is the room air should be around 21%, and you can see down here in the bottom corner, or if I want to turn on a meter, it's reading about 21.23%, which is a little high. That's okay. What you want is to make sure that it's actually reading, around 21, 20.9%, something like that, what it should. If it's reading low, if you plug in any oxygen gas sensor, the O2 BTA, the older black one with the cable, or this one that's reading low, the cell may need to be replaced. And the cell probably needs to be replaced about like once every 4 to 5 years. That's covered under warranty. We do have cells that last a lot longer than that. There's some great tricks about getting the cells to last longer that I can tell you about if you want. And then even if it's out of warranty, you can get the cell replaced for about half the cost of the probe. So I'm going to calibrate the sensor here just to show you, hit calibrate. I know that should be around 21%. This relative stability reading down here tells you what it's reading. That means that's what it was reading when it was calibrated. So it's basically at 100% of what it was when it was shipped. So I know that I have a good cell. For the GoDirect sensors, if that number is low, that tells you that it's bad. And you can see that this was calibrated in 11/8/2022. So I'm going to hit keep. Then I'm going to hit apply. Great. So my O2 gas sensor is calibrated. I'm going to go back here and I'm going to pop it in the top of my chamber, stick my CO2 gas sensor in there. That's 389 again. All right. So let's connect the CO2 gas sensor. There we go. I saw some chats come through. Don't worry, I'll get to those here in a minute. There, so I've got CO2 and O2 connected. I'm going to tap in the bottom right corner where it says sensor actions, and I'm going to change the units. I'm going to change one to PPT, change the other one to PPT. And you can see what it did is it came up with two graphs. So I've got O2 and CO2. And now I'm just going to wait for a little bit for everything to equilibrate, look at these chats, and then I'll hit collect. Oh, okay. Let's see. I'll tell you how the CO2 sensor should be calibrated, no problem. Thanks, Judy Day. And then we'll also talk about the O2 gas sensors. They should be fine if they've been around for a couple of years. Just get in touch. If you email biology@vernier.com or get online and go to chat, we can take you through if they need to be calibrated, what they are, but they should be fine even if they're a couple of years old. So here, let me hit the collect button. And we'll just take a look here and we'll look at oxygen gas and see if it's going down. It's going down slowly. CO2 is going up. So let's just do that little magic trick of zooming to all data. You can see O2 is going down, CO2 is going up. There we go. I mean, that's the key thing we want to show, right? In this experiment is that O2 is going up, O2 is going down, CO2 is going up. Cellular respiration is a real thing. Calibrating the CO2 gas sensor is pretty straightforward. You should take it outside and it should be reading around 400, 410 ppm, around 400. If you need to calibrate it, the trick is you just take it outside with the little Nalgene bottle and capture some CO2, some air from outside with the CO2 sensor, bring it back inside, let it sit until it comes to room temperature, and then just go through the calibration process. If you have our GoDirect CO2 sensor, you want to tap on the meter and calibrate it. If you have the older one, what you want to do there is just gently tap in on the calibration button and let go. You just want to tap in and let go, and it'll start blinking red, and that tells it it's at 400. All right, so there's O2 gas going down and CO2 going up. Gosh, it sure would be nice if we didn't have to wait 5 minutes for this experiment, right? Well, we don't, because this experiment also lives in GA Pro as a video. Right. Oh, and right. So if I want to, at the end, if I want to look at the respiration rate, I drag across, look at the respiration rate, etc. Look at a linear fit. The O2 should be calibrated at around 20.9. It depends. You could say 20.9, you could say 21. Either one is going to be fine. If you look in the manual, it'll actually give you an adjustment that you can use for relative humidity, temperature, and things like that. So you can say 20.9, you could say 21. Especially for a lot of experiments in biology, what we're trying to do is see if they go up or if they go down. Oh, how does it differentiate between photosynthesis for O2? It doesn't. It's just measuring oxygen consumption, right? The peas, though, are not going through photosynthesis at all here. There's no light. They don't have any leaves. There's nothing like that. That's what's really cool is if you actually look at spinach leaves. If you look at spinach leaves in the dark, you'll see cellular respiration, right? You'll see CO2 go up. And then after it's gone up, if you shine a light on it, you'll see all that CO2 then disappear. It's getting sucked back in. So that's how you would not be able to differentiate specifically between cellular respiration and photosynthesis using the oxygen gas sensor. It's that you're overriding one or the other. When you have photosynthesis going, you're overriding cellular respiration. All right, so to see this experiment in real time, or not in real time, but a video of it at the end of the experiment, I can also go to new experiment, don't save. Actually, hold on here. Let me disconnect these two channels again. But I mean, you saw that that's real data. O2 goes down, CO2 goes up. Not to, as would be expected, CO2 looks a lot better than O2, but the O2 level does go down. CO2 level does go up. So I'm going to say don't save. Go back to C experiments. And we can take a look at CO2 and O2. And in this case, what you can see here is non-germinating versus germinating. I'm going to turn off non-germinating. I'm going to turn, there we go. So you could see, let's turn off non-germinating. So same experiment, same setup, great data, right? O2 declines, CO2 goes up, which is exactly what you want to see. And the trick there, the key things are make sure your O2 gas sensor is calibrated. And then also make sure you use a lot more Ps. Use like 60. Fill the bottom of this BC250, this biochamber, with Ps. You'll get it to work every single time. Questions? No? Everybody with me? Everybody still here? All right. So I want to show you one more really cool experiment in the time that we have left. Yes. Great, thanks. I want to show it to you because I think it's really neat. I think it's really cool. It's very different. But it's just another way to look at cellular respiration. Don't forget, if you have any questions about how to make any of this inquiry, please ask. There's a whole lab that looks at sugar metabolism. So it uses the CO2 sensor, and you look at how the CO2 sensor how yeast can metabolize different types of sugars. Great lab, perfect for inquiry. Takes a little long to set up, but once you have it, students get it going. It's great. All right, so what I'm going to do is I'm going to show you first. What I'm going to do is show you the experiment that lives in GA Pro, and then we'll go back and I'll show you the experiment in the video, or actually in live first. So let me go to new experiment, don't save. There is this really cool experiment that exists in Biology with Vernier. It's called aerobic respiration. I don't know that a lot of people do it. I just wound up doing it for fun. It works really well. So I'll show you the basic setup here. So it's a small beaker of 4% sucrose and water. And now we've got a temperature probe here, but what we forgot is the dissolved oxygen probe, the GoDirect dissolved oxygen probe, has its own temperature probe. So you don't even need this temperature probe to do this. And if you take a look here, all that it's doing is you're just spinning it here. I'll show you what the experiment looks like really quick. Here, let me enable replay. And what's great is this experiment, you look at room temperature water, you look at cool, and you look at warm. So you look at the respiration rate of yeast in water in 3 different conditions: room temperature, cool, and warm. So let's just show you room. And I've sped it up. You could play this faster. I put in 4 mils of yeast suspension. We begin collecting data, and you can see up here what happens is, right, the dissolved oxygen level starts to decrease. That's the yeast consuming the oxygen that's in the water. And it happens actually pretty fast. I mean, you could see it happen within like 20, 30 seconds, you'll see it decreasing. So I like this experiment because you don't even need peas. All that you need is a little stir station, you need a dissolved oxygen probe, and you need some yeast. The key thing with the yeast that I have found is you need to make sure you have yeast that haven't expired, and you can't just keep them on the shelf for several years. You probably want to buy a fresh packet of yeast. And the other thing is make sure you activate them with water that's warm. So that's all in the teacher's instructions, but when you get it started, just make sure the water is warm-ish, like 35 degrees, not over 40, but just make sure you use warm water. But this is a great demo that I can get going anywhere when I'm doing a workshop or a training, because all that I need is a stir station, a dissolved oxygen probe, a little plastic beaker, my little stir bar, and just some yeast. 1 packet of yeast, thrown in 100 mils with a packet of sugar. That will work for the entire classroom to do this experiment. And then what's great about it is I can also take a look at, it's got really good results here. So if you look at cool versus warm, I mean, you can see if you were just to take a look at the respiration rate here, let's turn on the graph legend. There you go. So you can see that for warm, less dissolved oxygen will be in the water to begin with, because it's warm, but the rate, like the slope, is much more negative, right? So it's consuming more oxygen. If you look at cool, starting around 40 seconds on, the slope there is less. And if you look at room temperature, the slope is intermediate. So you also get great data if you want to show someone, or talk about how for an organism like this, if it's warmer, it's going to consume more oxygen. If it's cooler, it's going to consume less oxygen. Oh, what temperatures do you use on the cool and the warm? I don't think it's that critical. It just, in the teacher's instructions, which I can give you, they're there. You just don't want it to be like over 40 degrees or boiling. Actually, I can see, I think it actually just tells you what they are. Hold on here. I can tell you specifically what they are. Give me a minute. Aerobic respiration. Does it even say? Let's see here. Oh yeah, for cool is 10 to 15 degrees. And for room temperature, it's between 20 and 25. And then warm is 30 to 35. Don't go above like 37 degrees. So that's a, not a bad temperature range. I don't even really need to use ice. It's just a really nice, cool little experiment. So questions? Go back to my presentation. I mean, I can show you the live demo of it. I just want to show you, this is, the GoDirect dissolved oxygen probe. Let's take see what it looks like. I'll move this out of the way here. It's a probe, looks like this. It is rather expensive, but it's incredibly useful. Once again, USB port. The temperature compensation is right down here on that little metal port. Let's see, I've got a chat. Yeah, please go ahead and take the survey, give us some feedback, because I've got about 10 minutes before we all take off. So yeah, we went through that live demo, and I mean, I pretty much have shown you all of the key things that I wanted to show you, which are the GoDirect CO2 gas sensor, how to use it for cellular respiration, the O2 gas sensor, and then the GoDirect optical dissolved oxygen probe, which is great. I have everything here prepped and ready to go to show you, just so you can see that it's not that hard. Let me just do that really quick. There you go. So I just have, a little beaker of sugar water, some sucrose and yeast. So if we had time, I was going to show you that experiment too, but it doesn't look like we do. So what I'll do is I'll stop in the time that we have left. And we can get some questions asked. Just to review, let me review the two books there. So there's Biology with Vernier. The 3 experiments that I went over were 11B and 11D. That's cellular respiration with P's, CO2 and O2, and then aerobic respiration. But there are 1, 2, 3, 4. There are at least another 1, 2, 3, 4 experiments just on respiration that you could look at, in photosynthesis that you could look at that use the CO2 sensor or CO2 and O2 gas sensor. The inquiry biology book is great. There are 3 key experiments. Part B, experiment 9, experiment 10A, and experiment 12A. This one's kind of interesting because it looks at evolution, the evolution of yeast, but it's just looking at the respiration rates as well. All right, so let's take a look here. All right, so let me answer your questions here. The price estimate on the dissolved oxygen, yes, I can. I'll show that to you next here in a minute. Oh, the wired. So if you have the wired optical DO probe, it's fine. It'll do fine with this, and it does have automatic temperature compensation as well. You can't see the temperature; it doesn't have a temperature sensor, but it is temperature and pressure compensated. Like I said, that probe will work great for this lab. You don't have to worry about it. It's great. It'll work terrific. There you go. So let's just take a look at these sensors here. So dissolved oxygen, if we go to products, go to sensors. Here are the GoDirect sensors. There's the GoDirect oxygen gas. It's around $200. The GoDirect optical dissolved oxygen is $359. And then there are these in alphabetical order. There's CO2 gas, $225. But once again, if you have the older probes, they can all be used. If you actually go in and you look at the book instructions, so if you have the book, buy the book, there'll be 2 sets of instructions for graphical analysis in it. One for what we call LabQuest sensors, and one that we call for GoDirect sensors. So all of the experiments that I've done today work great with the old sensors too. All right. Other questions, issues? Anybody want to talk about inquiry? Have other things that they would like to address? We have a pretty cool biology GoDirect package, which contains a lot of these probes, but like I like to say, Vernier is for teachers by teachers. You don't get a special deal if you buy the standard package. So if what you want is just CO2, dissolved oxygen, and O2, you can buy those a la carte. And you can also just buy the BioChamber 250 a la carte. Don't forget that the CO2 and O2 gas sensors are already going to come with an Nalgene bottle, so you don't need to buy that. We like to make sure that our probes come with most everything that you would need to do basic experiments with them. All right. And with that, I'll stop at any other questions. We've got like 5 minutes left, I think. Oh, great question. What is the average lifetime for the probes? It really depends. So the CO2 gas sensor is what we call solid state. should last a long time. The CO2 gas sensors that are used at Wartburg College, where I used to teach, are probably still being used since it's an IR column. So they probably will last a long time. We have a 5-year warranty on all of our probes. Most of them have the standard 5-year warranty. So that one's going to last the longest, provided you don't duck it in water or something like that. They do have a battery, and the battery in them, a little lithium-ion battery that can be replaced. And that might need to be replaced 3-4 years. And that isn't covered under warranty like after 2 years, but I have probes that I have used for over 5 years that are fine. I haven't needed to replace the battery. The dissolved oxygen sensor has a cap, and that cap will probably need to be replaced every 2 years or something like that, 2 to 3 years. It just depends on use. The oxygen gas sensor is the one that really will degrade over time. And that's why we make it so that you can replace the cell inside of it. One thing you can do that I've heard is if you place it in an area with like low oxygen, like I had a teacher that actually put them in a plastic food container where they sucked all the air out of it. And her oxygen gas sensors lasted like a super long time when she did that. I can't guarantee that they will last a super long time, but that is one thing that I saw, because her O2 gas sensors were over 10 years old. And I told her they were all dead and they were all fine. And then I said, how did you store those? And she showed me, oh, I've got, when I have steaks, I put them in here and I suck all the air out and then I seal it. Well, I do the same thing to the oxygen gas sensors. And I said, keep doing that. So you can try that if you want. You're not going to break anything. Just make sure that the oxygen gas sensors are stored upright. That's really important. The other thing is make sure the dissolved oxygen probe, for it to work well, it needs to be exposed to water. It needs to be in a humid environment. So just make sure the sponge is damp when you store it, or if it's been stored over the summer, it's gotten dry. Put some water on that sponge. Let it sit overnight. The probe, in the little container that it comes with on the tip, and let it just be in a wet, moist environment, and then it should be fine. You can always recalibrate it. All right. Well, I think that is it. Sorry, hold on here. That's how to use GA Pro. We're not going to go over that. Yeah, that's all I've got for you. Unless you have more questions or want to talk about anything, don't forget that you can always get a hold of us by emailing biology@vernier.com. You can find us on chat just by going to the website and going to chat. You can look for Sarah, Colleen, or Melville. Those are the 3 biologists. And you can also just call. We like talking to customers. Remember, Vernier is for teachers by teachers. We love talking to teachers. We love solving problems. So feel free to get in touch. All right. And thank you very much.
Thanks, everybody. Have a great rest of your day.